All right. Can somebody confirm you can hear me?
I can hear you.
Thank you. Up there? Yeah, I do. All right, great. Before we begin, are there any questions? Before I show my screen, let me talk a little bit about the grading. I know I got the last lecture graded. Once you post your answers to the lecture worksheet, you should check your posting to see if somebody else beat you to the question. Because to get full credit, you need to have your question not be a repeat of somebody else. So you should always check yourself after you post your answer. Um, anyways, worksheet for lecture 6 was graded. And then I think his lab 8 was graded. Let me check on that. Yeah, I got lab 8 graded for the first time. Few people got fives, but most people missed something. If you didn't get 100%, remember you have until I think it's this Saturday. Let me check on that. You have until May 24th to um, redo Lab 8 and then turn in your answers again. It'll be for full credit. All right, any question about any of that? If not, let me get out of the grade book. Close that down. Oh, and remember to work on your unknown project. I got answers up. I think I did it on Sunday and Monday. I meant to do it on Saturday and I never got it done. So, um, but I did get the answers up on Sunday, which I normally don't work on Sunday, but since I was unable to do it on Saturday, I did it on Sunday. All right, any questions about anything? If not, let me share my screen. You should be seeing my screen now. Let me remind you, tomorrow, this Wednesday, you have a quiz 4, and it'll be covering Lab 7, chapters 5 and 6. Today, we're going to talk about Lab 9, identification of gram-negative organisms. Any questions about any of that? All right. Lab 9, identification of gram-negative bacilli. Um, in this lab, you need to complete Lab 9. You need to complete the case studies and the worksheet for Lab 9. There is some information to read, so please read about the biochemical tests and bacterial identification. Only about a page, 131 to 132. And then in chapter 10, read about the methods for classifying and identifying microorganisms, about 4 pages beginning on page 272. There are some video clips in today's lab. Upon successful completion, you should be able, one, be able to interpret the results for the following biochemical tests for the IMVIC reactions. IMVIC is an acronym for the indole test, the methyl red test, the Voges Prescure test or VP test, and the citrate test. We're also going to talk about carbohydrate fermentation, which I think we've already talked about, but we'll talk about it again. We're going to talk about the motility test and the hydrogen sulfide production test. Any questions about these tests? You also need to be able to explain how to interpret the TSI test, which is the triple sugar iron slant, the SEM deep test, which is the sulfur indole motility test, and then the test of growing organisms on McConkey's agar and HE or ectone enteric agar. And then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. Any questions about these learning objectives? All right, we're going to be talking about the family Enterobacteriaceae. The members of this family are all small, gram-negative, non-spore forming bacilli, and some of them are so small they're almost caca bacilli, but mostly they're called gram-negative bacilli. They are all facultative anaerobes. There are 52 genera in the family Enterobacteriaceae, so it's a very large family. 24 of the genera are known to cause human infections, and all the members of the family are glucose fermenting and oxidase negative organisms. Here are some species or genera that commonly cause a disease like Salmonella and Shigella. Most of these genera are normal microbiota of the intestinal tract, meaning we call them coliforms. They're opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera responsible for opportunistic infections include Escherichia, such as E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera in the family Enterobacteriaceae are frank human pathogens causing gastroenteritis or some other disease, including Salmonella, Shigella, and E. coli Serovar 0157H7. Normal E. coli does not cause gastroenteritis to humans. In fact, normal humans have E. coli in their intestines, so we actually need it to help us digest our food. However, there are certain variants of E. coli, such as Serovar 0157H7, which is a human pathogen causing gastroenteritis. 0157H7, these are two antigens which normal E. coli does not have. There is also the Pseudomonaceae family, particularly Pseudomonas aeruginosa. The members of this family are similar to the family Enterobacteriaceae in that they are gram-negative bacilli. However, the Pseudomonaceae are all non-glucose fermenting. They are all oxidase positive, and they are also all aerobes. The Pseudomonaceae can be opportunistic pathogens, particularly immunosuppressed individuals, burn patients, and trauma patients, and that's mainly Pseudomonas aeruginosa. But Pseudomonas aeruginosa is also a health-acquired infection or a nosocomial disease. Any question about any of that? It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on non-gram-negative bacilli cultivated from clinical specimens. We're going to talk about some of the tests that you can do to identify the species. Like here's a large variety of tests that you could run to identify gram-negative bacilli. And here's some more, not as many, but some. We're going to talk about some of these, including the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added to the supernatant or the solution, Kovacs reagent will combine with indole to produce a red color at the surface. E. coli is positive for indole production. So here we have tryptophan, that amino acid, in the presence of tryptophanase, that enzyme, produces indole and pyruvic acid. And that's supposed to be a reddish color. This photograph is a little bit bluish and it's looking purple, but it's definitely not negative, which would be a yellowish color. Or somebody called it a light tea color. That person must be, they like their tea light like I do. But normally it's kind of reddish. It's not purple like that. Normally, this photograph is a little purple. Any questions about the indole test? All right, E. coli is positive for indole production. Off the top of my head, I can only think of one organism which is negative, and that's Micrococcus luteus. I'm sure there's many organisms which are negative, but off the top of my head, I don't remember them. And now let's talk about the methyl red and the VP or Voges-Percure test. This test is started by, or these two tests are started by inoculating one tube, which is the MRVP media. And the methyl red test looks at the production of acid products formed from glycolysis of glucose. And the VP test looks for neutral products from glycolysis of glucose. Any questions about that? Normally what you do is you inoculate one tube, incubate it for 2 to 5 days, depending on which test you're running, and if you're running both of them, you would probably do it for 5 days. And then you divide that tube into two parts, one part for the MR test, the other part for the VP test. Any question about that? Methyl red is added to the aliquot for the portion of the tube for the MR test. If bacteria produce acid, the pH indicator will remain red. And then the test is said to be MR positive. If the methyl red, when added, quickly turns yellow, then the test is negative. So there's MR positive, it's red, there's MR negative, and it's yellow. And the other half of the tube, which we use for the VP test, you add Barrett's reagent A and Barrett's reagent B, and then you shake the tube to add oxygen, and then you let the color develop for 60 minutes. Acetone, in the presence of Barrett's A and Barrett's B and oxygen, will turn red. So if acetone is produced, a neutral product from glycolysis, it'll turn red, and that's VP positive. You'll note that not all of the tube is red. Part of the tube is yellow, part of the tube is red. That's normally what happens in the VP test. And then VP negative, the entire tube will be yellow. Any questions about that? Now, the easy thing is that for both the VP test, the MR test, and the indole test, for all three of these tests, a positive result is red, a negative result is yellow. Any question about any of that? All right, the last test in the IMVIC series of tests is citrate utilization. Citrate is an important molecule in the Krebs cycle. If the organism can perform aerobic respiration, it'll engage in the Krebs cycle. And if it can utilize citrate as its only carbon source, it will grow in the citrate utilization test. And the tube will turn blue, it'll have growth on the tube. The growth isn't shown here, but it'll turn the tube blue. And that will be citrate positive. The blue color comes from the raising the pH in a basic, changing the media pH to a basic pH, and it'll turn blue. If the bacteria cannot utilize citrate, it will have no carbon source, so the bacteria should not grow. And the tube should remain green. All right. E. coli and Shigella can metabolize citrate, so they'll turn the tube blue. And once again, one of the ones I know off the top of my head is Micrococcus luteus will be negative. There's many others which are negative. I just don't know them off the top of my head. But Micrococcus luteus will be negative. It will not grow on citrate utilization test, and the tube will remain green. All right, any questions about the IMVIC test? If not, let's talk about the PR carbohydrate metabolism test. Carbohydrate fermentation is used to determine whether the bacteria have the ability to hydrolyze, meaning metabolize, a particular sugar. We have different sugars added in the carbohydrate test, and it's called the carbohydrate test, but it could be called the glucose test, the lactose test, the sucrose test, the maltose test, and practically any other sugar test. So when you look at the results I sent you for the lactose test, and you look up on the class website, it won't be under the lactose test because I've called them all. Now, where the heck is it? Oh, PR. It's under the PR. PR carbohydrate test. PR for phenol red. That's the pH indicator. And you can use one sugar in a tube, and then you just look to see if the bacteria can ferment that sugar. And there are some of the sugars, and it could be any other carbohydrate as well put in here. Anyways, if the sugar can be fermented by the bacteria, the acid products will be made and the pH of the media will turn yellow. If the sugar is not fermented, it will either remain red or become a pinkish color, and that will be PR negative. This reaction is not considered PR carbohydrate positive or glucose positive. This is considered A for yellow, meaning A for acid being produced. If, on the other hand, the sugar is fermented and the sugar is also producing a gas when it is fermented, we score this as AG, meaning A for yellow, and then G for gas being produced. And you can see the gas as a bubble in the inverted Durham tube. There's the inverted Durham tube. It should have media in it. And then if gas is produced, you'll see a bubble in it. No gas there, no gas there. So this one would be, if we had glucose, the sugar in these, then this would be PR glucose negative, PR glucose A, and this one would be PR glucose AG, for A for acid and AG for acid and gas. Any question about any of that? Like I stated with this test, you can have one sugar in the tube, and then you run the test for that sugar. So you could have glucose, you could have lactose, and I just call them all the carbohydrate fermentation test, but you could call it the glucose fermentation test or the lactose fermentation test or the maltose fermentation test or the sucrose fermentation test or any other sugar. Any question about any of that? All right. This test is looking at the motility test. If the bacteria are not motile, then they will only grow in the stab. So this is a semi-solid agar. It has very low agar, and that's why it's called semi-solid. And the bacteria cannot, well, they can swim in it, but if they're not motile, they'll only grow in the stab mark. And so that would be motility negative. If they're motile, and especially they're highly motile, then the bacteria will swim everywhere, and you can notice cloudiness throughout the tube. Let me blow this up a little bit. Right here, you can look through there and you can see it's not cloudy, and there it's cloudy. So there's a stab mark there. You can't even see it because the bacteria is growing in the stab mark and everywhere. So that's very motile bacteria because the whole media is cloudy. If a portion of the media is cloudy, then it's motile. And if it's slightly motile, it won't grow only in the stab, but it won't be cloudy like that. Let me show you what it'll look like. It will be growing in the stab mark, but it'll be flaring out from the stab. And let's make the media here. Some of them will be symmetrical, some of them will not be symmetrical. And that's what a flare would look like. Any question about any of that? If it's flared, that means it's slightly motile, but it is motile. And that's showing you highly motile. Any questions about the motility test? Remember, if bacteria are motile, they have at least one flagellum on the cell. They may have many flagella on the cell. To do the motility test, you have to be careful when you stab it. You have to stab straight in and pull it out straight out. Because if you were to go in at an angle, and then, I don't know if you can see this, pull it out at another angle. So you're going in at an angle and move it over and then pull it out. It will look like it's at least flaring. And what you can do is usually rotate the tube and you'll notice it's not really flared, that it's just a bad stab mark. All right, let's talk about the hydrogen sulfide production test. Some bacteria can produce hydrogen sulfide gas. We have an ingredient in the media which can act or react with the hydrogen sulfide gas and make a black precipitate. You can test for hydrogen sulfide production in the TSI test. That's a triple sugar iron test. You can test for it in the sulfide indole motility test, and you can test for it in Hectone and Terra agar or HE agar plates. All of them, the bacteria will look black, forming a black precipitate if the bacteria can produce hydrogen sulfide. The family Citrobacter, Salmonella, and Proteus produce hydrogen sulfide gas, and they will stain black in one or the other. That looks like this looks like the Sims media test. and you can see black usually only on the bottom, although this is almost all the tube, but the black usually shows up at the bottom of the tube. You can also get it to show up on the triple sugar iron slant. Let me show you that here. So there's black from hydrogen sulfide being produced. Once again, you find it on the bottom of the tube. And there's another Sims tube with black on the bottom of the tube. And then here we have a hectonenteric agar. These colonies are black. That's from the production of hydrogen sulfide gas. These colonies are not black. That's because they're not producing hydrogen sulfide gas. Any questions about hydrogen sulfide? And sorry, we do have 3 tests that you can use to look for hydrogen sulfide production. Let's talk a little bit about the triple sugar iron agar slant. This is a slant shown here. But when we do this test, we inoculate the slant, shown here, as well as we inoculate the butt, shown here. In fact, you should go down more. This is only going down about halfway in the butt. You should go down about 2/3 of the way in the butt. And the reason for going 2/3 of the way down is that the black usually forms at the bottom of the tube. It does not form near the top of the tube. And that's because hydrogen sulfide gas is usually made when the organism is not performing aerobic respiration, and many organisms will perform aerobic respiration if they can, because that's the most efficient way of making ATP. And so the top of this tube will not be black, but the bottom of the tube, which here would only be right here. And that's why you should go down about 2/3 of the way. You should not go down all the way because you don't want to damage the needle on the bottom of this glass tube. Anyways, you inoculate the butt by putting bacteria on the needle. Let me show you this. Hopefully you can see this. So let's pretend this is our colony and this is a needle. You roll the needle in the colony and then stab the needle. Obviously, you have the needle sterile. Then stab the needle into the butt. This will inoculate the butt. You then take the needle out, streak it through the colony again, and then you inoculate the slant of the TSI. The purpose is to get the bacteria to grow in the air on the slant and then to grow at least partly anaerobically in the butt. Any question about any of that? All right, the TSI slant has 3 sugars in it, glucose, lactose, and sucrose. But note the glucose is at 1/10 the concentration, the lactose and the sucrose. It is a red, phenol red pH indicator. That way you can look to see if a sugar is fermented or not. It has peptone for a source of nitrogen. And then importantly, it has sodium thiosulfate in it. And that allows the detection of the hydrogen sulfide gas. If hydrogen sulfide gas is produced, it'll react with the sodium thiosulfate and the ferrous sulfate to produce a black precipitate, which will be black like that then. Similar, that's the wrong two, but all right. The results will be multifaceted. You have to look at what's happening on the slant. So this would be negative on the slant because it's the same color, no change. Negative in the butt. So that would be, actually that could be K over K, meaning alkaline over alkaline. You could also mark it as, and I normally use it as negative over negative, meaning negative on the slant, negative in the butt. No gas is produced, no hydrogen sulfide is produced. For tube KA, We'll talk about the slant in a minute. Maybe I should go to tube 4. It's a little easier to talk about. For tube 4, it is A over A because we have acid being produced on the slant. So that's the first A for acid. And then it's yellow in the butt. So that's another A. So A over A for the slant over the butt. It's yellow in both the slant and the butt. So we know that glucose is fermented because it's the base sugar. And at least lactose or sucrose or both lactose and sucrose are fermented. Is that clear? We don't know which or both is being fermented, but we do know that one of them is fermented. I'll explain why we know about that at least one of them is fermented, and that's because it's yellow in both the slant and the butt. We then notice that there's cracking of the agar and there's an air bubble here, so you can say gas is being produced in the fermentation process. And it's not black, so there's no hydrogen sulfide being produced. So that explains tube 4. Let's move on to tube 3. On the slant, it's red, but in the butt, it's yellow. What has happened is that only glucose has been fermented. Why it is yellow in the butt, because there is one of the sugars that's being fermented. We know it's not both sugars because the slant was initially yellow. If you had looked at it earlier, it would have been yellow, but with time, all of the glucose got used up in the slant. And then the bacteria, rather than dying, started using the protein in the media. When you use protein, you make ammonia, and ammonia is a base, and that will change the slant from what used to be yellow to pink. And so that's, I'm going to call it negative. You can call it K for alkaline in the slant over A in the butt. And we know only glucose is fermented because glucose is metabolized slower anaerobically in the butt and it's metabolized quicker in the slant. And so all the glucose got used up in the slant. And then the color reverted to pink or red because of the ammonia being made by the bacteria digesting the protein. And I know that's a lot, but that's how you explain tube 3. Tube 4, we know that tube 2, we know that hydrogen sulfide is being produced because it's black in the bottom. If it's black in the bottom, then you know the bottom is also yellow, even though you can't see the color because yellow is being hidden by the black. But wherever there's hydrogen sulfide being produced, we know that there is fermentation, and so that would be A for the bottom, the butt. Hard to see the acid butt, but if it's black, you know it's fermented and it's then an A. There's no gas being produced because there's no cracking of the agar, there's no bubble. And here we once again see the slant is red and the butt we're assuming is yellow. You can't see it, but if it's black, then there has to be fermentation, so it's yellow. And only glucose is fermented and the slant reverted from yellow to pink because of the buildup of ammonia from the bacteria utilizing the protein instead of the sugar, which there was no sugar in the slant. There's still sugar in the butt. Any questions about any of this? So interpretation of TSI slant is multifaceted because there's a lot going on. We have growth on the slant, growth in the butt. We're looking for the production of hydrogen sulfide gas, no hydrogen sulfide gas. We're looking for the production of gas in the fermentation process, and you can determine that by a bubble or by breaking of the agar. And then we're looking at if glucose is fermented, if there is fermentation anywhere on the tube, it's safe to say that glucose is fermented because glucose is the base sugar. It'll always be fermented if another sugar is fermented. If both the slant and the butt are yellow, then glucose is fermented in at least one other sugar, either lactose or sucrose or both of them. We cannot tell if which, but at least one other sugar, if not both of them, are being fermented. And if only the slant is red and the butt is yellow, then only glucose is fermented. Glucose is used up more rapidly in the slant because sugar will be used by aerobic respiration, the most efficient way, the most quickest way to get rid of the sugar also. And in anaerobic conditions, the glucose will not be used up as quicker, at least in the time span of this test. And so the butt will be yellow, the slant will be red. Any question about that? All right. Let's talk about this SIMS test. This is a sulfide indole motility test. It's looking for the production of sulfur, hydrogen sulfide gas. It'll turn black. It is also looking for the production of indole, what the I stands for. You inoculate this, and then you take it out of the tube. You then add Kovacs reagent. It'll be floating, and so it'll be added to the top of the tube, meaning it'll stay at the top of the tube. If the tube turns reddish, this is kind of pinkish, but reddish, pinkish, then indole is produced. If it remains yellowish, that one's more yellow, then indole is not being produced. And then we're looking at motility. So this also has the motility test, meaning 3 tests in this one tube. If the organism only grows in the slant, like tube 2, then the organism is not motile. If the organism flares out of the slant, or most of the top of the tube, and then there's flaring out here, then the organism is motile. It can swim in the tube. Tube 5, it's only growing in the slant. It's hard to see tube 3, but it's only growing in the slant. And tube control is an uninoculated control, so there's no growth. Any question about the SIMS tube? SIMS test? All right. So it's looking at hydrogen sulfide production, looking at indole production, looking at motility. And then there's McConkey and HE agar plates. You can look at whether they're gram-negative. If they grow on McConkey's or HE or hectonecteric agar, then it's likely to be gram-negative. Gram-positives tend not to grow on McConkey's or HE plates. If there is growth on the plate, you can then determine whether the sugar lactose was fermented or not. A lactose fermenter on McConkey's will be pink colonies, and then the plate agar will be pink around the colony. So this streak here is pink. So this is a lactose fermenter streaked on this half of the plate. The bacteria streaked on this half of the plate is a non-lactose fermenter. It's gram-negative because it's growing on the plate, but it's not pink. In fact, that's white or off-white. And so it's a non-lactose fermenter. For the HE plate, if it grows on the plate, it's gram-negative. If the growth is salmon colored, that's a yellow-orange color, that means it can ferment the sugar lactose. It's salmon colored. If it doesn't ferment the sugar lactose, the bacteria will be their normal color, which in this case is white or off-white. Non-lactose fermenter. This is a non-lactose fermenter with the production of hydrogen sulfide gas, and it will be black because of the production of hydrogen sulfide. I would assume if the bacteria can ferment the sugar lactose, but also produces hydrogen sulfide gas, the colonies would be black, and you could not see the color in the colony, but you may be able to notice the orange color around, meaning the media might turn orange or salmon colored. Like right here, there's not growth, but the media is orangish color. So you might be able to tell lactose fermentation from the color of the agar around the bacteria, if the bacteria is producing hydrogen sulfide gas. Any questions about that? All right. Shigella can grow on HE or MAC plates because it's a gram negative. It can ferment the sugar lactose. So on HE plate, it will be, no, excuse me, on, I have to look that up. I think that's wrong. I think that's not on an HE plate, that it's blue, green in color. It'll be the color of the bacteria if it can't ferment the sugar lactose. And then a salmonella, which can ferment bac well, salmonella will produce hydrogen sulfide gas, so it'll be black. Any questions about any of that? All right. So you're not going to perform any of the activities in this lab. Everything will be provided for you, but you are responsible for knowing how to perform and interpret these tests. You can view the following video clip on carbohydrate fermentation, on the SIMS media test, on the MRVP media test. So that's two tests in one. It's one tube, you inoculate, but you split it into two, one for the MR portion of the test, and then the other portion of the tube for the VEP test. And then there's a video on TSI test, a video on McConkey's agar test. The Microbuzz website does not appear to work, but you don't really need it. Everything you need will be supplied in this lab module. And you'll need the lab module to answer the first part of the worksheet. So question 1, the oxidase test, the positive result has been done for you. You can't get this from this lab, but you can get it from an earlier lab, and that is the oxidase test. The spot turns blue, or the cells turn blue if you put it on the cells directly. The negative result, no blue. And then give me the results for the positive for indole and the negative, the positive result for the methyl red and the negative, the positive result for the VP and the negative, and the positive result for the citrate test and the negative. For indole, methyl red, VP, and citrate, all the results come from this lab. For carbohydrate fermentation, we've got the results in this lab as well. Give me the positive result and the negative. This is only looking at the sugar being fermented, like glucose being fermented. And then give me the gas being produced, what the positive result would be if gas is produced, and then in the negative result. So remember, you're not filling in the carbohydrate fermentation here. You're only talking about whether the sugar is fermented and whether gas is produced or not. And then give me the results for the triple sugar agar slant test. What is the result if glucose is fermented? Positive result, and the negative result. Remember, in the glucose fermentation of the TSI slant, you need to state the slant over the butt. So you should have two results in each cell, meaning if glucose is fermented, it should be A over the slant and A over the butt. Well, actually, if it's only glucose, it should only be actually negative in the slant and positive or A in the butt. And then for the negative result, it'd be negative over negative. If lactose or sucrose is fermented in addition to glucose, which that's not stated there, but if lactose is fermented, then glucose will also be fermented because the glucose is the base sugar. Give me the results in the positive case. Once again, that should be the result in the slant over the result in the butt. And then the negative result, which would also be negative for the slant over the results for the butt. And then for the TSI agar slant, give me the positive result if hydrogen sulfide gas is produced, and give me the negative result. And then if gas is produced, give me the result for TSI positive and the result for TSI negative. Remember, this is for the TSI slant, so we're not looking at color here. We're not looking at the production of a bubble in an inverted Durham tube as well. The triple sugar iron doesn't have those things. Actually, you can look at color, I guess, but that's not gas produced. That's the sugar being fermented. And then give me the, for the Sims agar deep test, give me the sulfide production, whether it's positive and whether it's negative. Give me the indole production if it's positive and if it's negative. Give me the motility if it's positive and if it's negative. I probably won't differentiate between slightly positive and greatly positive. And then give me for the selective and differential media, that's the McConkey's and HE. What is the positive result for lactose fermentation? And what is the positive result, negative result for lactose fermentation? Remember, the color will be different on McConkey's and HE if it's positive. And then, am I on the worksheet? Complete the following for 2A. 1. What reagent must be added to detect the presence of indole? If indole is present, the test will show what color. So that would be 2A2. Any question about any of that? For 2B1, methyl red is a pH indicator that remains what color under acidic conditions? And methyl red pH indicator is used in what test? Can anyone suggest that now? It's really easy. Question is, the methyl red pH indicator is used in what test? Think about it. That one's so easy. I'm not going to give you extra credit. All right. 2C, what test uses Barrett's A and Barrett's B reagent to detect the presence of acetone? A neutral product formed from glucose metabolism. If acetone is present, the test will show what color? So that would be 2C2. For 2D1, Simmons citrate uses what pH indicator? Name it there. And it turns blue under bold or acidic conditions. You can either write it there, you can circle it, you can bold it, you can underline it here. It doesn't matter. as long as you have an answer. But you do need to pull an answer for 2D2. 2E1, what is the pH indicator used to detect PR carbohydrate fermentation? Actually, that's what PR stands for, the pH indicator, by the way. And it is yellow under bold or right one, acidic or basic conditions. And you could highlight also. For 2F, bacteria possess this structure, a cell structure, that tests positive for motility. So you need to name a part of the cell, a cell structure. 2G, 3 organisms that produce hydrogen sulfide are, name the species or genera. 1, 2, 3. And it's given in the lab module. You can look online as well. If an organism ferments glucose but not lactose or sucrose, the TSI slant will be what color? And the deep butt will be what color? 2I, state the 3 tests for which SIMS is used. So SIMS stands for something. So state the first test, second test, third test. 2J, MAC and HE plates are both what? In that they inhibit the growth of a species of gram-positive or gram-negative bacteria. So you must bold or write or highlight gram-positive, gram-negative for 2J2, and then state what they are for 2J1, what type of a media it is. And then they are also what? In that they separate various bacteria based on visual differences from sugar fermentation. Any question about 2? For 3, we're looking at case studies. You want to use this key to identify the case studies in this lab only. Do not use another key. Obviously, don't use this key for your unknown project because it's only looking at 2 families, Enterobacteriaceae, and it's only looking at a few families of the family, meaning a few members of the family, because there's 53 genera, and I don't know how many species, probably over 200 species of bacteria in the family, Enterobacteriaceae. And then it's looking at Pseudomonaceae family, Pseudomonas here. So use this key to identify the bacteria of the case studies. And then use this table for case study 1. Let's go back to the lab module. Case study 1, a prison inmate is brought into the ER wound with an open wound. This prisoner is known for self-inflicting his wounds. A routine culture and sensitivity test is ordered. The appropriate transport media is used for specimen collection. There we're growing up the bacteria on the blood agar. You can note the hemolytic pattern. There's the organisms growing on McConkey's plate. And then the color of the bacteria. And this is the gram stain. To see that bacteria, you got to blow it up really, really big to see the shape as well as the color. Actually, the color, you probably don't need to blow it up too big, but you need to blow it up really big to get the shape. Oop, too small. Anyways, those are the initial results. We then do some rapid tests on day 2. We look at the catalase test and the results are catalase positive. The oxidase test is oxidase negative. We also run the SIMS media tube test, shown in A, the MR test, shown in B, the VP test, shown in C, the Simmons citrate test, shown in D, and then the triple sugar iron shown in E. So put your results in the table here. And then use the results for case study 1 using this key to identify the species. Case study 2, Mikey talks his mom into buying a pet turtle. I got to get to the results. And he's good about feeding and changing the water for the turtle, but he's not good about washing his hands after handling the turtle. 2 weeks after Mikey gets the turtle, he develops diarrhea, nausea, and fever. His pediatrician orders a fecal specimen. There's the culture of it with the arrow pointing at something interesting. And that's on a McConkey's plate. And the arrow pointing at colony of interest in the HE plate. You note the color on the HE plate. The results of the catalase test, the bacteria are catalase positive and they're oxidase negative. And there's the SIMS tube, the MR test, the VP test, the Simmons citrate test, and the TSI test. So once again, write your results for case study 2 in this table here: oxidase, glucose fermented, lactose fermented, TSI, whatever. And then use your results in this table with this chart to identify the bacteria causing Mikey's diarrhea and nauseousness. Any question about any of that? For case study 1, state the species that is causing the patient's symptoms. Here, for case study 2, state the species causing the symptoms here, and then do that for case studies 3 through case studies 5. Any questions about any of this? All right, I'll be here until 8 o'clock to help you with the lab if you have any questions. But once the last student logs out, I will log out as well. So if you have questions, you want to make sure you ask them while I'm still logged on. Any questions? All right. That's it for the lab. Get started on the lab.